2. I wanted to start today just by giving you an overview of the schedule for the week. So you have some idea of kind of what's coming and what to expect from our live sessions. Monday and Tuesday will be pretty basic. There will just be a short lecture with a little bit of group discussion in some breakout rooms. Tuesday, I will be introducing a project that you're going to work on, looking into a specific example of cell communication. And I'll give you a little bit of time in a breakout group with your partners for that project to figure out exactly how you want to break it down and how you want to work on it. Wednesday, there's no live session. You will just be working on your research project that day and finishing up mastering biology if you need to. There will not be a College Board assessment this week. Thursday is probably going to be the longest, most interactive class session. We're going to be doing a case study. So it'll be a lot of working in breakout groups and kind of discussing through an example of cell signaling. And so if you can be here Thursday for the live session, that would be really helpful. Also, Friday, we're going to be doing those group presentations for the research project. And so if you can be there to present with your group, that would be good. If you absolutely can't, we will talk about that. Contact me and we will make arrangements. So that's the plan for the week. Today we are going to continue working through the nervous system. On Friday, we talked about resting potential and how that's created and maintained in a neuron. Resting potential, if you actually measure the voltage, is between negative 60 and negative 80 millivolts, which means the inside of the cell is negative compared to the outside. And if you remember, this is due to a balance of chemical and electrical forces. Both the chemical is that concentration gradient, and the electrical is the attraction of the positive and negative that causes the sodium and potassium to move in and out of the cell. To create this situation where there's more sodium outside the cell, more potassium inside the cell, but overall the inside of the cell is negatively charged compared to the outside of the membrane. And that difference in charge, that membrane potential, is a source of potential energy. Just like a battery has stored energy because it's positive and negative on the two ends, a neuron has stored energy because of that voltage across the membrane. That allows neurons to send signals. When a neuron is activated, when you touch something or when you smell something or see something, or when your brain needs to tell your muscles to move, the neurons along that pathway get activated. And what happens is there's a shift in that voltage. So the negative and positive changes so that it's either more different or less different. And that difference in voltage causes some of the protein channels in the membrane, some of those ion channels, to open. So if you think about protein structure, when the voltage shifts, really what's happening is positive and negative charges moving around. Remember, a lot of protein structure has to do with interactions between positively and negatively charged R groups or hydrophilic R groups that are attracted to positive or negative charge. So if you change the positive and negative charge around a protein, it's going to cause all those R groups to kind of move around in response to that. You could kind of think of it as turning around a magnet. It's going to make any other magnets nearby shift position in response to that. And that causes these proteins to change shape in such a way that they open a channel through the middle to allow some ions in or out. Now, you don't need to memorize the details of potassium coming out and sodium coming in, but you do need to understand these terms hyperpolarization and depolarization. So hyperpolarization, you can see on this graph here, so this is resting potential. So this would be where that negative 70 is. A hyperpolarization makes the inside more negative. So hyper means more. So you're increasing the difference between the inside and outside of the membrane. And you can see here, because it becomes more different, it moves it away from what we call a threshold. We're going to come back to that idea in a minute. You can also have the opposite happen. If sodium flows in, then your positive charge is coming in. That leads to something called depolarization. So again, if this is our resting potential down here of negative 70, you can see when this happens, the inside of the cell is becoming less negative. So the inside and the outside are coming closer together. It's depolarizing. There are lots and lots of these stimuli coming into neurons all the time, depending on the signals they're receiving. And these are just small signals that vary with the strength of the stimulus. They don't last very long. But the thing is, if a neuron receives enough of the signals to reach that threshold, then it triggers something called an action potential. And so you could conceive of this kind of like this. So here's our -70. Here's our resting potential. And here, this line across here is not actually necessarily 0. This is what we call the threshold. This is the point where the voltage is different enough, where the voltage reaches a certain point. It's changed significantly enough that it's going to trigger an actual action potential, a signal to be sent by that neuron. So over time, you might get a little bit of a depolarization, a little bit of a hyperpolarization, a little bit of a depolarization. It might be going like this, but at some point, if you get enough signals that it crosses this threshold, this is what's going to signal that action potential. So remember, in your neuron, these are all of the dendrites, these little branches out here. This neuron might be receiving one signal here, a different signal here, a different signal here, and some of these are hyperpolarization signals and some are depolarization signals. So the neuron takes all those signals in, they travel through the cell body, and right here at the axon hillock, if you remember that term, it's kind of where the axon itself begins. This is where the decision, if you want to think of it that way, kind of the molecular decision is made whether an action potential is going to be sent down that neuron or not. It basically takes all of these incoming signals and adds them up like we just saw on this graph. And if all of these depolarizations and hyperpolarizations together add up to reaching this threshold right here, that is what's going to trigger an action potential. Oh, hang on. There we go. So an action potential is kind of an all or nothing thing. These hyperpolarizations, depolarizations can go up and down, but once you cross that threshold, an action potential is going to be sent down the neuron with the same speed and the same strength every single time. The way that an action potential works is it's a series of opening and closing of different ion channels, and you don't actually need to memorize all of these steps. What I want you to remember, I'm just going to show you this brief video clip, and I'm actually just going to mute my speakers because you don't need to hear this. I just want you to watch for a couple seconds here. Let me get this to the right spot. After a brief commercial, I want you to watch what happens to the charge along the axon here. And we're going to jump up to about 28 seconds there. We'll start at 25. So this is kind of a graphical representation of an action potential, but look at what's happening along the axon of that neuron as that action potential travels. It's a wave of changed voltage. You can see for a brief second the charge across that membrane kind of flips and that travels down the neuron. That's what an action potential actually is a change in that voltage due to the movement of ions on either side of that membrane. So as that signal travels, that's what's actually sending that electrical signal down the axon. It's important that these signals travel quickly because this is what helps you to respond to the information that your body's taking in and get away from predators or remove your hand from something that's hot or whatever you need to do. So there are some adaptations that help these signals travel faster. One of them is that if the axon itself is wider in diameter, there's less resistance. Just like a wider hose allows more water through, a wider axon allows the signal to travel faster. And we find this adaptation in some invertebrates. In vertebrates, the adaptation that helps us the most is something called a myelin sheath. You may have heard of myelin before. It works kind of like insulation that surrounds those axons. And the way that this works is the myelin sheath is made up of these things called Schwann cells. That's actually these things right there. That's the myelin sheath. And it kind of covers parts of the axon, which means the only places that those ions can diffuse in and out are these little places in between. They're called the nodes of Ranvier. It's a big fancy name for those. But the reason that this makes this go faster is that the signal kind of jumps from this open spot, leaps up to here, and then leaps up to the next one. And so they call this saltatory conduction. Those of you that have taken Spanish, if I remember right, the Spanish word for jump is something very similar to this. So that might help you remember what this means. So that myelin sheath helps that signal to travel more quickly. Now, when the action potential reaches the end of the neuron, it has to send that signal on to the next neuron in the series. If you look in the packet of papers I sent home that has your coloring page on the front, the next page is a series of pictures called How Do Neurons Communicate? Take a minute and look through those pictures and see if you can explain or take your best guess about what's happening in each of those pictures. So remember, we talked before about the fact that there's electrical communication down the axon of a neuron. That's that action potential and that change in voltage that we were just talking about. But that electrical signal can't hop across from one neuron to the next. And so to get from one neuron to the next, we need a chemical signal. So this is where we need some actual physical molecule to cross that space. The space between those neurons is called a synapse. And so when we're talking about a synapse, if we're talking about this synapse right here, we use the words presynaptic and postsynaptic to refer to the neurons that we're discussing. So if we're talking about this synapse right here, that's circled here, this is the presynaptic neuron because it's the one the signal's coming from. And this would be the postsynaptic neuron because it's the one that's receiving the signal. Keep in mind, those terms are just relative to this one synapse because then that chemical signal could signal another action potential, another electrical signal to go down here, which then would have to jump across the next synapse to the next neuron here. So if we were talking about this synapse between this neuron and the next one that's not drawn here, there we'll draw some dendrites. There's our next axon. There you go. If we're talking about this one, now this one would be the presynaptic cell. And this one on the right that I just drew in would be the postsynaptic cell. So pre and post are only in reference to a particular synapse. So when the action potential, that electrical signal, reaches the very end, that's called the axon terminal, it needs to cross that space. And you can see neurons are not actually physically touching. There's this little space here called the synaptic cleft. And so that signal crosses via chemical messengers that are called neurotransmitters. The way that works, when the action potential arrives at the end, so you have this electrical signal that's coming down this axon, that's our electrical action potential that we were just talking about. When it reaches the end, it depolarizes the membrane at the end, just like it's been doing all the time. So it changes the voltage around there, which opens some more ion channels. In this case, they're calcium channels that allow calcium ions to diffuse in. Calcium molecules or calcium ions act as a signaling molecule that then tell those vesicles to go and fuse with the membrane and release these red molecules, the neurotransmitters, into that synaptic space. In your note sheet, you have the next couple lines on this same slide. I just divided this into 2 slides, so you're going to see the same thing here. Those neurotransmitters are an example of a signaling molecule called a ligand. You don't need to write this down because we're going to talk a lot about this tomorrow, but I just want to mention it right now. A ligand is just a molecule that bonds to a protein receptor and causes that protein to change shape, which eventually signals something to happen in that receiving cell, in this case, the postsynaptic cell. So if we were to label these in this picture, this is our presynaptic cell up here. That's the axon terminal of the previous cell. That's where the signal's coming from. And this is our postsynaptic cell. This is a dendrite on the postsynaptic cell that is receiving this signal. And so those neurotransmitters, these chemical messengers, float across that opening and bond with a protein on the postsynaptic neuron. And you can see those are labeled ligand-gated ion channels. When they bond with that neurotransmitter, it causes that protein to change shape in such a way that it opens up. And that can lead to hyperpolarization or depolarization in that postsynaptic cell. And so that cell is receiving a signal that either makes it more or less likely that an action potential will start. The neurotransmitter itself can then be taken back up into the previous cell and put back into a vesicle and reused, or it can be broken down by enzymes. And there's a short video linked in the note here that can help you kind of visualize how this happens and how those neurotransmitters move across the synapse. Now, this type of signaling can be altered. These are chemicals floating through here, and so there are some things that can interfere with this process. A lot of drugs, the way that they affect you is because they interfere with neurotransmitters, either with the reception of that signal or with the ability of that signal to be destroyed. Sometimes they just increase the amount of a neurotransmitter, so your neurons keep receiving a signal and firing and firing and firing when they should stop. This can also be related to mental and physical health problems. So there are 2 videos that I'm going to link in the side here. Definitely watch the one that talks about cocaine interference as an example of how some drugs can interfere, like physically get in the way of this process, and how that can lead to addiction. There's another one linked in here, a TED Talk that's just really interesting and talks about how neurotransmitters and brain chemistry is related to things like behavior and ADHD. So if you have time, that one's a little bit longer, but it is really, really interesting. Tomorrow, we're going to focus a lot more on this idea of chemical signaling and how cells receive a message like a neurotransmitter and how that affects what happens in that cell. So we're going to be focusing on the details of that process a little bit more tomorrow. Feel free to contact me if you have any questions. Hopefully, we will see you at the live session tomorrow. Have a great afternoon.